Melanism, also known as melanosis, from Greek, dark-colored, is the occurrence of an increased amount of dark pigmentation, as of skin, feathers, eyes or hair in an organism, resulting from the presence of melanin. It is the opposite of leucism and albinism, which occur because of a lack of melanin or other types of pigment. Abundism is the occurrence of excessively abundant dark markings, such as spots, stripes or other patch types, due to increased dark pigmentation on the coat, fur or skin of some animal species. As the number and or size of these markings increases, some overlap and merge, causing the effect of pseudomelanism, where the animal appears completely black. The background color may still be discerned between the markings. Melanism and pseudomelanism is often the result of genetic mutation, but can result from other stimuli, such as exposure to abnormal temperature changes during gestation, which transiently alter gene transcription or translation. Melanism has been found to be linked to beneficial changes in the immune system. The Smithsonian Answer Book, Cats notes that genes for melanism in felines may provide resistance from viral infections, and that a viral epidemic may explain the prevalence of black leopards in Java and Malaysia, and the relatively high incidence of black leopards and black servals in the Aberdares region of Africa. Citation needed. Previously, black-furred felines in the Aberdares had been considered a high-altitude adaptation due to absorbing more heat. Citation needed. Studies reported in New Scientist magazine in 2003 also suggested that recessive gene melanism is linked to disease resistance rather than altitude. According to Eduardo Iziric and Stephen O'Brien of the United States National Cancer Institute in Maryland, the melanism mutations involve the same gene family as those involved in human. Melanistic cats may therefore have better resistance to disease than cats with normal color coats. This would explain why recessive melanism persists when melanistic individuals are disadvantaged due to being poorly camouflaged in open areas. In the United States National Cancer Institute studies, black cats were found to have changes to a gene known as MC1R. MC1R is a member of a family of genes that includes the human gene CCR5, which codes for a protein on the cell membrane. Melanism could make black cats less susceptible to certain viral infections, making melanism an evolutionary advantage. Researcher Sidney K. Pierce said the super-green sea slug, Alesia chlorotica, which lives in waters on the east coast of the USA and Canada, is known to steal genes in photosynthesizing organelles called chloroplasts from its favorite intertidal algae species, Vaucheria aurea, but it now seems it has developed an entire chemical pathway to manufacture the green pigment, chlorophyll A itself. Chlorophyll is the pigment that captures energy from sunlight in photosynthesis. Pierce and his team used radioactive tracing techniques to determine the slugs were manufacturing the chlorophyll themselves, and it did not originate in the algae they ate. A number of animals, such as corals, host microbes in algae and benefit from their photosynthesis, but in most of these associations the cells remain whole. In Alesia chlorotica, in contrast, the cells are broken down and chloroplasts are extracted and held inside the slug's own cells, where they remain active for the slug's lifetime of almost a year. Researchers have shown that once a young slug has eaten a meal of Vaucheria algae, it never has to eat again as long as it has access to the light and supplies of chlorophyll and other chemicals used in photosynthesis. In 2007, scientists, including Pierce and his team, found genes related to photosynthesis in the slugs, and these genes, apparently originally from the algae, were even found in unhatched slugs that had never eaten algae. In the latest research, Pierce found more algal genes, and some of them were for enzymes required for the chemical process manufacturing chlorophyll. Pierce and his team studied slugs that had not eaten anything for at least 5 months and had stopped eliminating waste digestive products. They contained chloroplasts taken from the algae, but Pierce said that any other part of the algae should have long ago been digested. They gave the slug an amino acid labeled with radioactive carbon and found that the radioactive carbon turned up in the chlorophyll molecule after the slugs had been sunbathing, but not if they had been in the dark. The findings were reported at the annual meeting of the Society for Integrative and Comparative Biology in Seattle, Washington on 7 January and will be published in the journal Symbiosis.